Hello everyone, my name is Elena Shekova and I'm an Early Career Fellow at the Emerson Center for Medical Mycology at the University of Exeter. First of all, I would like to thank organizers for giving me the opportunity to present my work at this conference, and I'm really grateful for this chance. Today I will be speaking about mitochondrial reactive oxygen species and how they regulate macrophage antifungal responses. I study redox biology of fungal infections, and in particular I want to understand how reactive oxygen species mediates the interaction between human fungal pathogen Aspergillus fumigatus and host. And actually we know for quite some time that ROS are important for antifungal defenses. For example, in 2003 it was shown that NADPH oxidase, the main producer of ROS in immune cells, was necessary for effective killing of Aspergillus fumigatus. This was shown on alveolar macrophages. These cells were treated with the TPI, which is the inhibitor of NADPH oxidase, and this treatment resulted in the reduced ability of immune cells to eliminate the Aspergillus canidia. This result was supported by in vivo data in which animals with defective NADPH oxidase had troubles to eliminate inhaled canidia as well. This is in accordance with the fact that chronic granulomatous disease, which is characterized by defective NADPH oxidase, is also a major host risk factor for the development of invasive aspergillosis. So what do ROS do to play such an essential function against pathogen? To answer this question, we will have a look at macrophages, and macrophages are among first cell types that meet Aspergillus at the site of infection. And what is happening is that Aspergillus canidia are engulfed by macrophages, and then NADPH oxidase complex is assembled in the phagosomal membrane. This complex is responsible for conversion of oxygen into superoxide, which can be further transformed into hydrogen peroxide due to the activity of superoxide dismutase. And we know that hydrogen peroxide, as well as other oxygen and nitrogen species, are able to inhibit the growth of Aspergillus fumigatus, although I have to say that Aspergillus fumigatus has quite sophisticated antioxidative systems. Then NOx-derived ROS can mediate redox signaling, and this is happening by modulating the functions of redox sensitive molecules such as proteins. And finally, NOx-derived ROS can also induce LC3-associated phagocytosis, an important component of macrophage antifungal defenses. While we are talking a lot about NOx-derived ROS, we are forgetting about another very important source of ROS in immune cells, and this is mitochondria. Indeed, we have quite a lot of mitochondria per one cell. It can be ranging from 80 to 2000 mitochondria per cell, and in immunology it is well appreciated that the function of that mitochondria can serve various functions, and they are beyond energy production. For example, mitochondria can be responsible for ROS production in immune cells, and this is happening in a very specific manner following activation of the cells. So the question I'm asking in my research, do mitochondrial ROS play a role in antifungal immunity? But this question is quite tricky because there are a lot of different sources of ROS just within mitochondria. But one particular mechanism is often associated with immune signaling pathways, and this mechanism is called reverse electron transport, and I will introduce you to this process. So first, what you see is forward electron transport chain. And basically what is happening here is that electrons are moved through the complexes in this chain, and this is coupled to proton pumping, which in turn is necessary for ATP or energy production in mitochondria. However, it happens that electrons are moved in the wrong direction, in the opposite direction. There can be different circumstances for this. For example, this can happen because of the accumulation of succinate in mitochondria, and then the activity of complex 2 will be increased, and electrons from this complex will be pushed to coenzyme pool and then to complex 1. And these electrons will become accumulated in complex 1, resulting in the increased superoxide production because they will be picked by oxygen. And overall, this will lead to the increased levels of ROS produced by mitochondria. To sustain this process, there will be also accumulation of hydrogen atoms in the intermembrane space of mitochondria, and this will be associated with the increased mitochondrial membrane potential and also with the reduced ATP production in mitochondria. What is quite interesting that this reverse electron transport was reported to be quite important for different functions of mitochondria. First of all, it can be important for activation of macrophages during infection, but also at the same time after prolonged stimulation, for example with the bacterial product LPS, reverse electron transport can be quite essential to shift macrophages towards repair and healing. So in my research, I'm asking two main questions. Are mitochondrial ROS level increase in macrophages during infection with Aspergillus fumigatus, and whether this is happening because of the reverse electron transport? And second question, what is the role of this mito-ROS? To answer this question, we took macrophages and infected them first with resting canidia, but we couldn't see any difference in mito-ROS in contrast to control cells. However, following infection with swollen canidia, we definitely could see the increase in the accumulation of mito-ROS in the infected cells. Then we pre-treated macrophages with rotenone, and rotenone can prevent reverse electron transport, and indeed it could reduce the accumulation of mitochondrial ROS in these conditions, suggesting that here probably mito-ROS were generated via reverse electron transport. And the scheme of this method, pathway you can see here. And if it's true, if reverse electron transport is happening, then we are expecting to see the increase in complex 2 activity in the respiratory chain due to elevated oxidation of succinate, but also because superoxide is produced in the complex 1 in the respiratory chain. This can also change or reduce the activity of complex 1 because it can be inhibited by ROS generated at this specific location of respiratory chain. So we assessed the activity of complex 2 and 1, and interestingly, only in case of infection with swollen canidia we could see the increase in complex 2 activity, which corresponded well with our other data here. However, we couldn't see any significant difference in the activity of complex 1 of the respiratory chain. But also, as I mentioned before, if we expect to see reverse electron transport, then we also expect to observe the increase in proton accumulation in the intermembrane space, and this will also result in the elevated mitochondrial membrane potential. And we measured this parameter, and again, in case of the exposure to swollen but not resting canidia, we could observe the increase in mitochondrial membrane potential. So it seems that indeed mitochondrial ROS are generated in macrophages infected with Aspergillus via reverse electron transport. But what can be the role of this mitochondrial ROS? And again, to answer this question, we have to look at what is known about mitochondrial and antibacterial immunity. First of all, it is known that mitochondrials can exhibit direct antibacterial functions, and this can happen through different pathways. For example, toll-like receptors can induce generation of mitochondria-derived vesicles, and some of these vesicles contain superoxide dismutase. These vesicles will be accumulated in the phagosomal inside the bacteria containing phagosomes, and this will help to create oxidative stress for this microbe and help to inhibit its growth. But at the same time, through also toll-like receptors, it was shown that mitochondria will migrate very close to the phagosome, and then ROS from this organelle will be easily accumulated inside the phagosome and again creating oxidative stress for bacteria. Also, it was shown that beside direct antibacterial function, mitochondria can also control inflammation by altering their cytokine levels in immune cells. And here one of the examples for this process. So here mitochondria are generated, and then they can mediate the formation of disulfide bridges in NF-kappa B essential modulator, and this will activate corresponding signaling pathway. And in turn, it is also important for expression of pro-inflammatory genes. So now let's have a look if mitochondrial ROS are also important to eliminate fungus, and whether they can also control cytokine levels in cells infected with fungal pathogens. So here we took bone marrow derived macrophages and applied RET inhibitors for 1 hour, and then the cells were inhibited, and we checked whether fungal growth was inhibited and whether RET inhibitors made any difference on this on the capacity of macrophages to do this. So here you can see that interestingly, both compounds which have a potential to prevent reverse electron transport could also reduce the ability of macrophages to kill swollen conidia or Aspergillus fumigatus. However, these two compounds are metabolic inhibitors, as they can have off targets. That's why I also applied mitochondrial antioxidants, which cannot affect oxidative phosphorylation, for example. And only the compound which can reduce reverse electron transport or prevent reverse electron transport, it could also reduce the ability of cells to eliminate Aspergillus fumigatus conidia. And interestingly, there was no effect from these compounds on the elimination of resting spores of Aspergillus fumigatus. So with this, we can definitely say that mitochondrial ROS in macrophages can contribute to the elimination of growing or swollen conidia of Aspergillus fumigatus. So what about cytokine secretion now? And interestingly, again, both compounds that can prevent reverse electron transport could also reduce the secretion of pro-inflammatory cytokine TNF-alpha, and the RET preventing antioxidant could serve the same effect as other RET inhibitors, and the same picture we obtained for another pro-inflammatory mediator, IL-1 beta. So with this, it seems that mitochondrial ROS is also important to mediate the cytokine secretion in cells when they're infected with Aspergillus fumigatus. But how do cells know that mitochondrial ROS need to be increased, and what is the signal for, like, upstream signal from mitochondrial ROS generation. And in the literature, there are a lot of reports suggesting that there is a crosstalk between NADPH oxidase and mitochondria, and they can control the functions of each other. And to test whether this is true, I thought that first it is essential to see whether NADPH oxidase phagosomal accumulation happens at the same time with mitochondrial ROS generation. Here you can see macrophages, which were infected with swollen Aspergillus fumigatus, and the jump in the phagosomal NOX2 enrichment happened after 1 hour of infection. And interestingly, the increase in mito-ROS generation happened at later stages of infection after 1.5 hours, suggesting that NOX2 phagosomal accumulation precedes elevated mito-ROS production in infected cells. All right, but what does it mean? Does it mean that NOX2 is important for the increase in mito-ROS generation? To test this, I took cells which are lacking NADPH oxidase, CGD cells, and quite interestingly, we could see that even at the resting stage, these cells have a lot of mito-ROS in contrast to the wild-type cells. And this was not very new, because in the literature there is a report on mononuclear cells from CGD patients, and in this report it was revealed that CGD cells have much more mito-ROS than wild-type cells. But what was quite surprising that these cells were not able to increase mitochondrial ROS generation when they were infected with swollen canidia of Aspergillus fumigatus, and then the rotenone also had no effect on these cells, suggesting that even though there are high levels of mito-ROS in NOX2 deficient cells, they are not coming from reverse electron transport. So let's take all of this information together. We know that NOX2 phagosomal accumulation happens at the early stages of infection, and this is important for the activation of reverse electron transport and associated redox signaling, which is necessary for cytokine secretion. And the treatment with rotenone can prevent reverse electron transport and thus reduce the secretion of cytokines by macrophages. However, when NADPH oxidase is missing, then RET cannot be activated, and cytokine secretion cannot be controlled via this pathway, and also rotenone will be ineffective. To confirm this model, we took NADPH oxidase deficient cells, and they were infected with Aspergillus fumigatus swollen canidia, and what is interesting that rotenone had no effect on these cells, as we, for example, saw with wild-type cells, suggesting that indeed our model seems to be correct. So with that, I would like to summarize. We saw that mitochondrial ROS are induced in cells infected with swollen but not resting Aspergillus fumigatus canidia, and probably this is happening because of the reversed electron transport. Then blocking this process can reduce cytokine secretion and also fungicidal activity of macrophages. And finally, NADPH oxidase is required to regulate cytokine secretion via RET-associated mito-ROS. So with that, I would like to thank people who were involved and contributed to this project directly or indirectly. So first of all, my mentors, Adelia, Al, and Gordon, then Remy and Arnav for their support. Liz and Guillaume for sending me tissues from CGD mice, and also Ivy, Fabian, Emily, Alessandra, Alex, and Christina for very interesting and always useful discussions. And some of these people are presenting posters, so please get in touch with them and discuss science that we are doing at the Emerson Center. And of course, thank you all for your attention, and I hope I will have an opportunity to discuss this project with you. Thank you.